This is a production of Cornell University.
So thank you for the introduction and the invitation to come. I've never been to Ithaca, so I mean, we're like the same state. We should have, you know, it's a very long state. But all right, so life on our planet has found ways to use roughly 1/3 of the elements that occur naturally on our Earth. And of these elements, my research focuses mainly on the first row transition metals and zinc. And what's really fascinating about these elements is that they provide nearly universal essential functional capabilities to enzymes and proteins. Usually they're needed in trace amounts, but there's some really fundamental functions that need these elements. I mean, what would we do without transcription, translation, respiration, photosynthesis? All of these processes and many, many more require one or more transition metals to function. And then what's really fascinating about these elements is if they're allowed to overaccumulate in the cell, it can actually be highly cytotoxic. And so because of this dichotomy between we need these elements, they're essential nutrients, but if they're allowed to overaccumulate, they become toxic. The abundance and availability of these ions in the cells are really tightly regulated. And organisms can be negatively impacted by both limitation, if there's not enough of these nutrients, you die, and they can be negatively impacted by excess. If there's too many of these metals, you die. But most of us in this room, we're not thinking, hey, did I get enough zinc in my diet today? Did I get enough iron? And that's because it's only when we see mutation in these homeostatic mechanisms that we see, oh, how important these nutrients are. And so a single point mutation in a metal transporter, for instance, can be really quite detrimental. And so metal homeostasis describes those strategies that evolve to ensure that supply of these metal nutrients meets demand. But you have to remember that this is a bit complicated because both the supply of these nutrients and also the demand for these nutrients can actually change. And there's some challenges. So one is that most metalloproteins, it's not all metalloproteins, but most metalloproteins need to bind the right metal. My favorite example of this is photosynthesis, electron transfer. And so what we see here is that the midpoint potential for these different metal cofactors varies over a volt. And that's because there's a really intimate interaction between the actual protein, the particular cofactor, porphyrin ring, iron sulfur cluster, and then the metal. And so here, for instance, we got 4 iron, 4 sulfur, 2 iron, 2 sulfur, hemes, copper, manganese, and really quite a breadth of midpoint potentials. Another great example here is plastocyanin and cytochrome C6. These are 2 soluble proteins that transfer electrons between the cytochrome B6F complex and PS1. These proteins are completely different proteins. They don't have any sequence similarity. They do the exact same reaction. They have the exact same midpoint potential. One uses copper and one uses heme to do that. Now, the other challenge is that actually selectivity is not very precise. And this can potentially lead to mismethylation. So you have these proteins, they need the right metal, but it's not that straightforward. This is the Irvin-Williams series that's been described since the 50s. And really what we see in chemistry is that all things being equal, if we look at these divalent metal ions, that generally, regardless of the metalloprotein, copper is going to bind with the highest affinity or the highest stability, followed by nickel, cobalt, iron, manganese. At the same time, a lot of these metals, if they're free, like if they're allowed just to float around in the cell, they can be toxic. So the metals that can actually transfer electrons can participate in Fenton and Fenton-like chemistry, and this can create oxidative stress, which can be very damaging. And then metals like zinc and copper can bind non-specifically to proteins and lead to protein precipitation. So the main question in my group is, how do cells transport and traffic these ions to ensure proper metalloprotein biogenesis while avoiding toxicity? And how I do that is that my group, we integrate a lot of genomics and post-genomic data, also mining high throughput experimental data to generate hypotheses about gene and protein function, and then we actually go to the bench and test And so to illustrate this approach, I wanted to go through 3 separate vignettes that are published and that I'm currently working on in my group. So the first is a copper chaperone for the chloroplast. All right, so copper. So copper is kind of at the top of this Irvin Williams series. And so it's thought that copper the cell is really careful about copper. And that once it's imported into the cell, we think that there's actually these small soluble copper chaperones. In some cases, we think they actually may dock with the copper transporter and deliver copper to where it needs to go. In eukaryotes, one of those is this copper ATPase that's in the trans-Golgi gene. And it's thought that this copper chaperone actually docks with this kinked helix. And so what this allows is direct ligand to ligand transfer of copper. So copper is never free. It's always bound to a protein. All right, so I'm interested in plants, green algae, plants, you name it. And so one of the questions I was interested in as a postdoc is, well, how is copper trafficked to the chloroplast? So in the chloroplast of plants, we have this plastocyanin protein. And this protein actually gets transported as a nascent polypeptide across the envelope and then the thylakoid membrane of the chloroplast. Once it's in the lumen, it folds, then it binds copper. And copper is the essential cofactor for the electron transfer. So that means that copper has to, once it gets into the cell, has to cross the envelope, the stroma, all the way down for maturation of plastocyanin. And some years ago, the group of Marinas Pilon actually found that there's 2 copper ATPases. These are the same family of the ATPases we find in the Golgi. And they pump copper across the inner envelope and the thylakoid membrane for maturation of plastocyanin. But the big question was, well, what about a copper chaperone? If you're ever going to need a copper chaperone, the chloroplast is the place where you're going to need a copper chaperone, because the chloroplast is actually making oxygen. Copper and oxygen do not mix, creates very dangerous reaction oxygen species. So what about a copper chaperone? So the first place is to start with the obvious. So the obvious would be to look for homologs of known copper chaperones. And you can apply a bit of logic and some rules so that the missing chloroplast copper chaperone should only be found in genomes with a chloroplast that has plastocyanin. We think that they should interact with the ATPase. So the ATPases PA1 and PA2 should be there. And then it should be conserved in all of those genomes. And this is basically just the green algal genomes in the land plants. And so this is a protein similarity network. Each of these dots here is a protein. The lines connecting them is sequence similarity between those proteins. And what you see here is that there's actually been a really large expansion of these copper chaperones in land plants. So which one is the copper chaperone? Well, if we include Chlamydomonas, which is a green alga, in this analysis, there's only one copper chaperone here. And this is probably the copper chaperone for the trans-Golgi, the known copper chaperone. So this isn't really a good bioinformatic hit for what's the missing chloroplast copper chaperone. So if the obvious isn't that obvious, what about something that's not so obvious? And this is what I found. This is looking at the gene for PAA1, which is the copper ATPase in the inner envelope. And what I saw is that this gene actually has an alternative splicing event that produces 2 transcripts. The first transcript, this long transcript, encodes the full-length copper ATPase. And the second transcript produces just the N-terminal metal binding domain of the copper ATPase. As soon as I saw this, I said, aha, this is interesting, because I knew already that the true soluble copper chaperones, the N-terminal metal binding domain of the ATPases, this domain of the copper chaperones are all structural homologs of each other. So a simple hypothesis is this the missing copper chaperone for the chloroplast? So in order for that to be the case, remember I had these rules, it should be here, not here. And so at first I was like, well, this is an alternative splicing event. Are alternative splicing events really that conserved across genomes? And the answer is, in this case, yes. So here I'm looking at, this is Chlamydomonas reinhardtii. This is the single cell green alga. These are various land plants. The evolution here is at least 750 million years. These guys have been diverging. And in every single case, I found evidence of a long transcript that's just the copper ATPase, and a short transcript that's just that N-terminal metal binding domain. When I was doing this analysis, I was really frustrated because there were some land plants, some genomes, I just could not find evidence of alternative splicing. And so I was really frustrated until I figured out that in a lot of genomes, there's actually been a duplication event of this gene and subfunctionalization, where now one locus has the full-length copper ATPase, and the other locus just has this short N-terminal metal binding domain. And then what's interesting is that we've kind of captured a snapshot where there's been independent duplication events leading to subfunctionalization. And then here in the grasses, there's actually been the duplication event was in an ancestor. And now you have two genes, one for the metal binding domain and one for the copper ATPase. All right, so let's test it. So we did a couple of different experiments. This is looking at copper transfer between this N-terminal metal binding domain and the transporter, the ATPase. And what we do in this experiment is we load the putative chaperone with copper, we mix it with the transporter, we then re-purify the transporter and ask, does the copper co-elute with the transporter, which would happen if there's been transfer from the chaperone to the transporter. And so here we're looking at the putative copper chaperone with PAA1, which is the transporter that it shares the gene with in a lot of genomes. And sure enough, we see co-elution between the transporter and copper, suggesting that we did see transfer. We can do the same experiment with PAA2, which is the second ATPase in the chloroplast, and we see no evidence of transfer. So it looks like it's specific to PAA1, not PAA2. The next thing we can do is we can actually, so both of these ATPases have their own metal binding domain. There's no evidence that the second ATPase, PA2, there's no evidence that its N-terminal metal binding domain ever exists as a free standalone protein, but we can clone it as a standalone protein and repeat these assays. And what we see with that is that for either PA1 or PA2, we see no transfer. So it looks like this is specific between PA1 and PA1's metal binding domain. These transporters are copper stimulated ATPases. They're ATP-dependent pumps. So we can also look at ATPase activity to test this interaction. So here are the two purified proteins where we're just providing free copper and measuring the ATPase activity. And then we can also provide copper already bound to the putative copper chaperone. And so we see stimulation of ATPase activity for PA1, not PA2. And then we can do the same thing with this internal metal binding domain of PA2, and we see no stimulation of ATPase activity for either transporter. All right, so in a nutshell, that's what I love to do. I love to look at comparative genomics, think about gene and protein function, contextualize it with evolutionary principles, come up with a hypothesis and test it. I put these arrows here because often when we get to the bench, the hypothesis is either wrong or maybe there's something different going on. One thing I'm not going to go through, but an example of this is with this story that we actually found that this transporter is actually flipped in the membrane versus how it is in bacteria. All right, so let's move on to heme. So iron. So iron, very important. Inadequate access to iron chronically limits photosynthesis, both on land and also in the oceans. And so some years ago, we found out that in some places in the ocean, it's actually iron that's limiting for phytoplankton growth, not nitrogen, as you would usually expect. It's also predicted that 30% of arable land, so this is our farmland, is actually considered iron limited. And then actually iron deficiency is the most common and widespread nutritional disorder in the world. In particular populations, children, pregnant women are particularly sensitive to iron deficiency. So iron is probably one of those micronutrients that some of us at certain points in our life really do need to think, are we getting enough iron in our diets? So I've been really interested in this question, which is how is iron trafficked to the chloroplast? As you can see here, the electron transport chain is really highly rich in iron, needs a lot of iron. And so one place to look is to look at phylogenetic inventories. And what this basically means is that you can generate lists of protein families that are only found in particular genomes from organisms with a specific phenotype, trait, you name it, whatever. And so in this case, I was interested in gene families that are only found in plants and green algae. So only found in those organisms that have photosynthesis, but also in the green lineage. And so these are some lists that have been around for a long time. These are called the green cut lists. And so these are inventories of proteins that are only found in eukaryotes with chloroplasts from the green lineage. And perhaps surprising, although these are kind of like those genes and proteins that make a plant, there's about 550 of these that we don't know what they do. It either has an unknown function or the function that's been annotated is vague and who really knows. So one of the projects I'm collaborating with, a big consortium of people, is to take lists like this. DNA synthesis has gotten pretty cheap these days. We're collaborating with JGI to synthesize and assemble these into vectors. I have a collaboration with Steve Almo at Einstein to do small expression screens to see if we can actually have protein to work with. And then once we decide we have protein we can work with, I do what I do with comparative phylogenomics and genetic hypotheses, and then we go in and test it. And so one of the proteins that I wanted to talk about today is this protein. So this green alga chlamydomonas, it's annotated as PPO3. So it's actually been given a name, even though we don't know what it does. It's annotated as this pyridoxamine 5' phosphate oxidase-like. In Arabidopsis, the ortholog of the chlamydomonas protein is annotated as FMN binding, which is a very specific and very vague annotation at the same time. Phytosome, which is my go-to database for plant genomes, it's annotated like this. This basically just means that it has similarity to a panther domain. So C-reg protein, okay, again, not too useful. All right, so warning about functional annotations. 1, the functional annotation is or could be wrong, okay? The annotation could be inaccurate, misleading, incomplete, vague. Okay, rule number 2, you need to know where the annotation came from, if you care, okay? And what that means is you need to actually understand which protein was actually experimentally characterized to have that functional annotation. And then the last rule is, again, if you care, which I care, you need to know how your protein, subfamily, or family of proteins is actually related to that characterized protein. All right, so where did these annotations come from? Now, it can actually be sometimes impossible to figure out where a functional annotation comes from. In this case, we can do a phylogenetic reconstruction of this superfamily. So this clade down here is the protein I'm interested in, this PPO3. Up here, this is the clade. All these stars are proteins that have been experimentally characterized to have this function, pyridoxamine 5' phosphate oxidase. So these are proteins from Arabidopsis, human, Drosophila, E. coli, Saccharomyces. So these are the true pyridoxamine 5' phosphate oxidases. C-reg, this one here. This is, again, an independent clade. This is the C-reg protein that's been characterized in human. Down here is my PPO3. It's actually more similar to this protein here called GLU-TRBP, which is a heme-binding protein that regulates glutamyl tRNA reductase, which is the first enzyme in tetrapyrobiosynthesis. And this protein family is more similar to that protein than it is to the pyridoxamine, the PNPOs. Something else is you can look at the domain architecture of these proteins. And so here they all have this split barrel domain here in blue, this whole family. GLU-TRBP and my PPO3 protein actually have a second domain that these proteins don't have, a DUF, which stands for domain of unknown function. If we take a closer look at these domains, so this split barrel domain family It's called the FMN binding split barrel domain superfamily. So now we know where that Arabidopsis annotation FMN binding came from. This is looking at the whole superfamily. It's a huge superfamily. Each of the nodes, which you can't see, actually represent more than one protein. This little tiny clade right here, or cluster, these are the true PNPOs. Down here, we have these C-reg proteins that are really only found in human, animals, and plants. And then this cluster down here is this tetrapyrrole biosynthesis protein, binding protein, and the protein I'm interested in, PPO3. We can zoom in. This is looking at those proteins in this cluster that have this DUF domain. And then something that was interesting when I looked at the superfamily is that there's actually another cluster of proteins over here, hug-Z, that also has this DUF. So the same architecture, domain architecture. If we look at just the DUF, so these are all the proteins that have this DUF domain here. So here what's nice is that yes, PPO3 is similar to this binding protein, but you can see that it's actually way more similar to a lot of bacterial proteins. So these are proteobacterial proteins, these are actinobacteria proteins here. Down here is this hug-Z. Something else that's interesting is that although this hug-Z, which is actually a bacterial protein that's been implicated in heme degradation and heme utilization, its domains are actually swapped versus the GLU-TRBP, which again is a heme binding protein. Within this DUF family, we also find proteins that look like they're heme oxygenases. So we have this DUF fused to what looks like a heme oxygenase domain. So a simple hypothesis here is this DUF a heme binding domain? Well, we can test that. So we purified the protein from this green alga chlamydomonas, which has the 2 domains. We also purified a protein from cyanobacteria that only contains the DUF domain, this DUF. And what we can see is that in vitro, this is a classic shift, the Sorey band here, that if proteins bind heme, you see a pretty characteristic shift. Not so prominent with PPO3, the cyanobacterial protein. You can see it quite clearly here. But of course, seeing is believing, and we were actually able to get some structures. So this is the structure of this DUF protein from Synechocystis as purified. And what we were surprised to see is actually the as purified form binds zinc and has a pretty, traditional zinc site. Here, a tetracoordinate zinc. Now, whether it actually binds zinc or not is a whole physiologically huge question, but looks like it binds metal. That's something I can confidently say. What was really interesting is when we added heme to the crystallization buffer, and what we found is that the dimers rearrange, and we get the craziest heme binding site I've ever seen, that anyone has ever seen, where we actually have 2 zinc sites. We have a doubly deprotonated histidine that's actually bridging the zinc and the iron in the heme. So it's kind of like a trinuclear metal site. Right now, I've contacted people who are supposed to be the top in the heme field, and they've never seen anything like this. So it's interesting. I have no idea what it does. However, it's not completely unprecedented. In copper zinc SOD, we see a doubly deprotonated histidine that bridges the copper and zinc. So having a doubly deprotonated histidine isn't too crazy. Also, there's a heme transporter in bacteria that has the iron in heme is coordinated by 2 histidines, and it's thought to protect the heme during transport. We also see this form in the hemochrome state of hemoglobin, and we also see this in the hexacoordinate hemoglobins. The issue is, though, is that in the hexacoordinate hemoglobins, the second histidine the binding is reversible, but those hexacoordinate hemoglobins, they don't have these additional metal sites. So the question is, what are these zinc sites actually doing? What I can tell you is we don't know. So, I'll have to leave it there. But what I can tell you is that this metal binding site, we really only see it in the cyanobacterial proteins. However, it's absolutely conserved in all the cyanobacterial proteins. Outside of cyanobacteria, basically half of this metal site's conserved. But it does look like that when we've had fusion with this oxidase-like domain, this split barrel domain, like we see in plants and like we see in the heme degradation protein hug-Z, it's coincident with loss of this heme site. So the summary for this part is that we found a new zinc, iron, heme trinuclear site. We couldn't have predicted this from the sequence. And we are working on answering some questions. One is these metal sites involved in heme binding. The most important question, is this structure even physiologically relevant? Does this zinc site form in the cell? And then we still don't know what the function of this protein is. All right, so the last story, real quick, is a universal metal transferase that has been my on and off obsession for some time. All right, so zinc. Zinc, very important. Zinc is involved in regulating transcription, RNA polymerase, carbonic anhydrases, the ribosome, protein synthesis, copper, zinc, SOD. Lots of proteins need zinc. It's important for immunity. Various neurological diseases, disorders have been linked to zinc. Cardiovascular disease, fertility, intestinal health, you name it, zinc has been implicated. So, there's all these important zinc proteins. Well, how are they actually loaded with zinc? As far as we know, we have a pretty good understanding of how zinc is transported across the membrane. It then enters a black box, which we call the labile zinc pool, which is basically just anything goes. It's probably some cysteine-rich proteins, metabolites, free amino acids, you name it, who knows? And then magically, proteins become loaded with zinc. Okay. And maybe that works as a model, that's fine, but what about under zinc limitation? What if there isn't enough zinc to go around to all these essential proteins? Does life have a way to actually prioritize that zinc so that the essential zinc proteins get zinc, but the non-essential zinc proteins, we say, we don't care about you, you don't get zinc. This is the screen alga chlamydomonas, which has been a really great model system for looking at metal limitation. And so this is looking at RNA-seq from various types of nutrient limitations. ZRT1 here is a known zinc transporter. And there's these two genes, ZCP1, ZCP2, that are really only expressed under zinc limitation. What's interesting is that in bacteria and in humans, homologs are also induced by zinc deficiency. Pretty much they all have unknown function. And eukaryotes and really bacteria, they're typically annotated as cobalamin and biosynthesis proteins, which in eukaryotes doesn't make a lot of sense because eukaryotes cannot make cobalamin de novo. So what are these proteins doing? So this is the family. So here I'm looking at the P-fam that detects these CobW-like proteins. It's a pretty big family. There's over 40,000 members. Archaea, bacteria, eukaryotes, they're all over the place. What's interesting right off the bat is that these proteins are related to UREG and HYPE-B, which are known GTP-dependent proteins that are involved in metallocenter biosynthesis of urease and nickel iron hydrogenase. Just to orient you, the two chlamydomonas proteins, one is found in this cluster, and then the other one is found in this cluster. If we look at the sequence similarity of these proteins, they have some very characteristic GTPase motifs. And so it looks like for the most part, this activity GTPase, and then also metal binding, the cysteine-rich motif, is conserved. And so a hypothesis really based on similarity to UREG and HYPE-B is that these are GTP-dependent metal transferases involved in metallocenter biosynthesis. For the ones that look like they're induced only under zinc limitation, it must be a function that perhaps becomes essential during zinc limitation. So maybe they're needed for delivering zinc or another metal to a zinc-dependent protein during zinc limitation. Now, but to test this hypothesis, we need the client protein. Now, the tricky thing with this family is that it's a really large protein family. There's multiple distinct clusters. Some organisms, like my favorite green alga, Chlamydomonas, have many homologs. There's more than 10 of these proteins. And then individual proteins from various bacteria have been linked to different functions. So down here, we have the proteins that were first characterized to be involved in cobalamin biosynthesis back in the 90s. These are the true CobW proteins. We also have a protein up here, NHA3, that's an iron-type nitrohydratase activation protein. And then up here, we have John Hellman's recently characterized ZAG-A. Well, it's not recently characterized, but the real big discovery for this protein happened recently. ZAG-A, which it looks like it's actually a GTPase and a zinc chaperone for this protein FOL-E. I've been really interested in what's the function of these proteins in eukaryotes. So this cluster up here contains the homologs from human, yeast, and plants. If we take a closer look at the specific cluster, so here is a phylogenetic tree looking at this cluster. And what we see here is that there's one clade here, so these are all orthologs that are found in eukaryotes, and they're typically only found in a single copy. And so one assumption is that likely these orthologs all share the same function, and maybe they all have the same client protein. So this is the protein that they deliver metal to. So one thing we can do is there's a lot of high throughput protein-protein interaction data out there, especially for these organisms. So this is yeast, Saccharomyces cerevisiae, Schizosaccharomyces, and then also human. But what you can see here, these are all these CobW-like proteins, which I'm calling GMC1. And these are all the interactions that have been found in these high throughput studies. But the question is, well, what of these interactions is actually biologically significant to my question at hand? A lot of these interactions could be experimental artifacts. They could be interactions that are important for regulation. I'm looking for a very specific interaction. So I looked at what protein-protein interactions are conserved across all of these very different organisms, and there's actually only one. This is methionine aminopeptidase type 1. It's a binuclear metal-binding protein, just like urease, which is the target for UreG. It has an essential function, and it's thought to be a zinc-dependent protein. But again, I wasn't convinced. It's like, it's high throughput data, is it valid, is it not? I like to look at the evidence. So if we look at the evidence, we see, okay, there's a yeast-2 hybrid experiment in Saccharomyces, another yeast-2 hybrid, okay, Schizosaccharomyces. In humans, there was a completely different type of experiment. This is affinity capture MS. And there were at least, 3 different studies that found this interaction. So it's like, okay. What was also interesting is that there's 2 classes of these methionine aminopeptidase in eukaryotes, a type 1, a type 2. The interaction was always with the type 1. But what really sealed the deal for me was this, that there was a high throughput genetic interaction study in Saccharomyces cerevisiae. And so if you knock out both MAP1 and MAP2, it's lethal. And they found a genetic interaction between GMC1, the putative chaperone, and MAP2, which is the second type of methionine aminopeptidase. So when I saw this result, I said, yes, we have to test whether this is the client protein. All right, so just this methionine aminopeptidase, if you don't know, this is a protein that cleaves the initiator methionine off nascent polypeptides as they're coming off the ribosome. It's a very important reaction. All right, so the hypothesis is that members of this particular subfamily in eukaryotes are metallochaperones needed for the biogenesis of type 1 methionine aminopeptidase, and this function becomes essential during zinc limitation. So we can design a genetic experiment where the model is that under zinc replete conditions, there's plenty of zinc to go around, MAP1 is metallated, no problem. Under zinc limited conditions, however, perhaps MAP1 is no longer competitive enough to interact with this labile zinc pool, but the model is that this putative chaperone is able to interact with this pool, bind zinc, and then in a GTP dependent manner actually transfer the zinc to MAP1. So what we can do is we can knock out the chaperone, and we would expect that under zinc replete conditions, the cell doesn't really care. But under zinc limited conditions, the cell is probably unhappy. But you have to remember there's this type 2. There's a little bit of functional overlap between type 1 and type 2. So the cells probably aren't that upset with the lack of this chaperone. But if we knock out MAP2, then the cells are probably really unhappy under zinc limitation. All right, so I decided to do these experiments in Saccharomyces cerevisiae because it's a wonderful organism. It only has one of these CobW-like proteins and only has one MAP1, one MAP2, so the genetics are very straightforward. And you just look at Saccharomyces and genes just pop out. It's great. So here I'm looking at YPD, which is a rich, undefined medium. This is looking at GMC1, MAP2. This is wild type, both genes present. Just GMC1 knocked out, just MAP2 knocked out, and this is the double mutant with both knocked out. And then here I'm adding increasing concentrations of EDTA. And so what I see is that right away, just knocking out GMC1 does lead to an EDTA sensitive phenotype that is exacerbated if we also have MAP2 knocked out. I've also, this is looking at a defined medium, where what I'm doing here is these are the same mutants, except I'm also bringing back the putative chaperone on a vector to see if I can rescue the phenotype with the gene in trans. This here is an empty vector. And so what we see here is if we reduce the amount of zinc in this defined medium, we see a little bit of a growth defect with GMC1 that's rescued, but we see a pretty drastic growth defect under low zinc concentrations if both GMC1 and MAP2 are knocked out. So this would suggest that GMC1 loss does lead to an EDTA sensitive phenotype. I think it's zinc specific that's exacerbated by the loss of MAP2. But the real key here is this is phenotypes, this is genetics, which that's my brain automatically goes, let's test this with genetics. But there's a lot that could be going on to explain those phenotypes. So we need direct evidence between the chaperone and the putative client. So I designed an experiment this is how it works. We have MAP1. If it's not loaded with metal, it's not active. We can then preload the GTPase with metal. We can then mix them together. Probably nothing's going to happen. But if we add GTP, then we see transfer of the metal, and we should be able to see MAP1 activity. All right, and so as some controls, as published, yes, we see metal-dependent activity of MAP1. No metal here. We can load it with various metals and see activity. The GTPase from yeast, again, this is pretty typical of the bacterial homologs, that it has kind of low intrinsic GTPase activity that's stimulated with various divalent metal ions. So this is our metal transfer assay. We purify these proteins, we remove them of all metals. We then preload the chaperone with metal, and then we combine the two proteins, and here we're assaying the methionine aminopeptidase activity. So this is just the methionine aminopeptidase by itself, no activity. You can add the chaperone without preloaded metal, no activity. But if you add the chaperone preloaded here, zinc, nickel, manganese, doesn't really seem to matter in vitro. We see activity of MAP1. This is with GTP. We can also add what's considered a non-hydrolyzable or slow hydrolyzable GTP analog, and we see no evidence of transfer in this case. And then what was exciting is as we were finishing up these experiments, John Hellman's group published this paper on ZAG-A and Bacillus subtilis, where in their case, which is probably really dependent on the client that it's interacting with, that it can actually hydrolyze ZTP. So we tested that in our system, and we do see some hydrolysis of ZTP. But when we do the metal transfer assay, ZTP is not able to catalyze metal transfer for our protein at all. So in conclusion, we think that the yeast GMC1 can transfer metal in vitro, and that this activity is dependent on hydrolysis of GTP. And I think what's also interesting is this starts to suggest some hypotheses about the mechanism of metal transfer. And so what I think is that the GTPase is switching between a high affinity metal binding site and a low affinity metal binding site, and that's what enables metal transfer. All right, so what about the chloroplast? So what's interesting is these in plants and algae, these are cytosolic homologs. I hypothesize that all of these are interacting with the cytosolic methionine aminopeptidase. This clade down here, it's only found in eukaryotes with chloroplasts. And what's interesting is that this protein does localize to the chloroplast in Arabidopsis. More than that, it's actually found in a large molecular weight complex in the stroma with MAP1B, which is the chloroplast localized methionine aminopeptidase. And something I was really surprised to see is that I went to ADDED, which is a gene expression database, and the top co-expressed gene with my chloroplast localized GTPase is MAP1B, which again is the chloroplast methionine aminopeptidase. And so we've actually tested this, and sure enough, we see that the chloroplast, which I'm calling GMC2, can transfer metal to the chloroplast methionine aminopeptidase. So this is our model. Like I said, we are thinking that it has a high affinity metal site that can interact and actually grab zinc from this zinc depleted pool. GTP hydrolysis allows switching to like a low affinity metal site, which enables zinc transfer. Of course, we need more experiments. All the experiments we've done so far do not tell us which metal is actually being transferred, because at least in vitro, there doesn't seem to be any specificity. And so we're working on assaying the actual activity in cells, and then also we have a collaboration to work out the different metal binding affinities in the high affinity form and the low affinity form. And with that, think my various funding agencies right now, Department of Energy, previously NIH for the copper chaperone, my postdoc Miriam, who's been doing all the metal transferase work, and Kumran, who is the crystallographer in the group for the heme binding protein. And with that, I'm happy to take any questions. Yes.
So if you jump to the heme binding group, if you provide reducing equivalence and redox partner, do you think it can act as a heme oxygenase, just like Vibrio HUD-Z or HUB-Z from strep?
Yeah, so we've done those experiments, and we've used ascorbate. We haven't used like a reductase. We've just, So if the domain is fused to this oxidase-like domain, it does look like it's a heme degradation protein.
Do you know your product for that?
We do not know the product. All I know is that the activity looks like it's very similar to HUG-Z. So even though the domains are switched, the activity is pretty similar. I don't know what what the product is, which is really interesting because this protein localizes to the chloroplast, and bilins are involved in retrograde signaling. There's all kinds of fun and wonderful stuff there. So yeah, that's a whole avenue of research that we're just not getting into. That's a completely new heme degradation protein in plants.
I'm just I'm just wondering, I don't know whether it's known or what are your thoughts about how chaperones and a whole bunch of different metal ligands in a cell, they do coexist, and with respect of metal transfer to transporters, and then versus short distance transfer and transport and long distance transport. Hoshiamine, histidine, glutathione. Yeah. How it all happens. What are your thoughts on that? Yeah, I know it's probably impossible to answer, but what are your thoughts on that?
I have no idea. So the question is that, so this is actually a really great question, because I think a lot of times in the metal fields, we study each metal in its own box. So we study copper, and then this lab over here studies zinc, and this lab over here studies iron, and we only ever think of one metal at a time. And we're not thinking too much about speciation and what it's bound to. So the question is, but in real life, all of this is happening at the same time. You have all the metals together, you have lots of different ligands, and then in plants it's complicated by, I'm focusing really on once metals are in the cell, but in plants we have root to shoot transfer. There's all kinds of really fascinating things going on, and how does the cell organize all of that when you have everything going on? Yeah, I guess I don't know.
I guess it's a fair question, but it's just like, yeah.
Yeah, I mean, I know at least in the case, like, because the other tricky thing is a lot of the divalent cations, especially in plants, are transported by divalent metal transporters, which it looks like do not have selectivity for one ion versus another. There's some selectivity with copper in that copper can be reduced to copper 1. And so transport by CTR, at least, is very specific to copper 1. So I don't have an answer for root to shoot transfer, but at least once copper is at CTR and comes into the cell, you could have selectivity that's mediated by protein-protein interactions. Because you have a transporter, you have a chaperone that's like literally delivering it to where it needs to go. You know, but I don't think people really know. Yeah, that's, yes.
Where does the complexation of the copper and the plasticinin take place?
In the thylakoid lumen.
So it takes place. So is there any effect of light-induced pH, the pH change, on the complexation?
Oh, I don't know. Yeah, I don't know. And because there's kind of an open question right now. So I didn't get into this, but the copper chaperone that I worked on, it looks like, because of the orientation of the ATPase, it looks like my copper chaperone is delivering copper to within the inner envelope space. So we have copper coming in, I don't know, somehow. The copper chaperone delivers it to the ATPase. We don't know how copper passes the stroma. That's an open question. Then copper is being transported by this ATPase in the lumen. I've seen some at conferences, I've seen some suggestion that plasticinin maybe docks with the ATPase, so there's direct ligand transfer from the ATPase to plasticinin. But whether effects on pH, I don't know the answer to that.
Did anyone look at glutathione? Because it's produced in chloroplast,
as like the chelate,
as a possible shuttle for copper within.
Yeah, I don't know. Yeah.
So PA1 and PA2, are they nuclear encoded or?
Yes.
So when the duplication or when the separation occurred of the metal binding domain from the PA1, how does it ensure that it goes into the stroma?
Yeah. So, okay. Yeah. So I didn't talk about this at all.
Because the localization signals are typically on the N-terminals.
Yeah. So there's definitely the signal peptide is retained after you have the duplication, and it's really clear because PA1 and PA1 and the chaperone, but not PA2, they have a signal peptide that's followed by a glycine-rich stretch. And we think that glycine-rich stretch is what localizes it to the inner envelope. So it's kind of like a stop transfer. And so whenever we see duplication of the chaperone, it maintains the signal peptide and this glycine-rich stretch. And what's interesting is that in the grasses, there's been further duplication of the chaperone. So it's like once the chaperone had its own gene, then the plants were like, oh, every time there was a duplication, they kept the duplicate. But in those cases, it lost that glycine-rich stretch. So perhaps that's neofunctionalization of copper chaperones, but they're not going to the same localization as the chloroplast-specific copper chaperone. Yeah, but whether the copper chaperone just exists in the inner envelope space and copper gets across the outer envelope, who knows? Maybe it's just bound to glutathione. It's a big open question. Yeah. Let's go here, and then we'll go this way. Yeah.
So, do you think the metal banding can help chaperone activity?
Whether the metal binding can help the chaperone activity? What do you mean? Like?
Yeah, I just wonder if there are some chaperone the metal banding will.
Oh, yeah. I don't know what effect, whether there's a pool of metal chaperones that are just in an apo form that are waiting for metals, or how dynamic that is. I don't know the answer to that. Yeah, I mean, for some of these, a lot of these, especially for like zinc and copper, there's cysteine rich. So there's a whole nother question about, because you can get disulfide bonds between the and like how that affects the regulation of these metal chaperones. Because if the metal's bound, those cysteines are protected from oxidation. So that's a whole nother, can of worms. Yep, Lars.
I was wondering, for example, the zinc chaperone, does it like deliver zinc to several other proteins, or is it like specific to one protein? And are there like chaperones for all these different metal?
Great question. The answer is I don't know. The second answer is I think so. Maybe it's not the case for all of them, but I think probably for some of them, I don't think it's unreasonable to think that they could have multiple clients. But then it becomes a question of, if you have one client and you can mediate metal transfer based on very specific protein-protein interaction, then that's a mechanism for ensuring that you're transferring metal to the right protein. If it was kind of a transferase that can interact with a lot of different proteins, then the question becomes, well, how does it become selective and specific for all of those proteins, but not all of these other proteins? Like, to my mind, it seems like it'd be a lot simpler to evolve a function that's specific between the transferase and one protein. But I don't think there's any evidence that rules out that it could be a more general function. Yes.
Thanks for your clear presentation. I'm interested in the second story, PPO3. The original annotation is a pre-doxamine related oxidase. It is a B6.
Yes.
And you just test the annotations the first by using the maybe comparative genomics.
Yes.
Reconstruct the gene family networks and test the function. It is involved with the zinc or heme bonding. So my question is, would it possible to test all the genes one by one, just using the computational biology approaches, no experimental or no bench work? If we have enough computing resource, can we test all the gene functions and improve the annotation quality of the genome?
So it depends on how much information you have about the experimentally determined enzyme. So for instance, with the oxidase that's been characterized to be involved in B6 biosynthesis, if you know the catalytic residues that are important for that reaction, you can ask, are those residues conserved in like other members of the family? And you can have a fairly confident hypothesis that based on conservation of those specific amino acids, that biochemical reaction, that function is conserved or not conserved. Because if those catalytic residues aren't there, then it probably doesn't have the same catalytic function. Yes, you can do that. The problem is that in a lot of the proteins where we know the function, especially enzymes, we don't have that level of information, that these are the catalytic residues that form that function. So the problem we run into right now is we're looking at the sequence similarity across the whole protein. And so there's issues there. One is that you can have two proteins that have 95% sequence similarity. One amino acid is changed, and now it binds a different substrate or has a different function, okay? Or you can have two proteins that you probably can't even tell they have sequence similarity if you just do a pairwise alignment, and they have the exact same function. So that's kind of what we're dealing with when it comes to biology, is you can have either or, and we just don't know. I mean, yes, right now, so if you look at the statistics, about 99.95% of proteins in our databases are not characterized, are not associated with experimental data, okay? That's way too many. I mean, that's like millions and millions and millions of proteins. We're never going to experimentally characterize everything. We're not going to do it. So we do need computational tools. But, you know, how do you get from there to there? I mean, yeah. There's just, I mean, with a more of a phylogenomic contextualizing type approach, you can at least kind of say, okay, this is the function, and maybe I'm moving more towards that's not the function, or maybe I'm moving more towards that is the function. And then if it's important to you, test it.
If there are no more questions, let's
This has been a production of Cornell University, on the web at cornell.edu.